Okay, good afternoon, everybody. Well, I guess it's still good morning. My name is Joe Panginis, Managing Director of Equity Research here at HC Wainwright. Very pleased to have a discussion today about the diabetes landscape, including Biomea's icovamenib's recent data at ATTD. And I've been calling this a late breaker KOL event to discuss these data. And very happy to have with us today Dr. Stephen Edelman. He's Professor of Medicine and Endocrinology at UCSD. He's also the founder of Taking Control of Your Diabetes, which was founded in 1995. Dr. Edelman is completed his diabetes specialist training at the Joslin Diabetes Center in Boston, came to the University of California at San Diego, where he's been on staff since 1990. He's an international leader in diabetes treatment, research, and especially education. He really champions the cause of patient advocacy and has dedicated his life to helping people with diabetes live healthier and happier lives. He's written 4 books and more than 200 publications under his belt, including major diabetes journals. And of course, that's only a small snippet of his very extensive CV and impressive CV. So Dr. Edelman, thank you very much for being here today, and I look forward to a very fruitful discussion.
Yeah, thank you, Joe. That was a very nice intro. I appreciate it.
No, thank you. So, I guess before we get into the real, context of clinical data, why not first get a little bit of your, little more of your background and your current research efforts in diabetes?
Yeah, well, as you mentioned, it goes way back, and I've been around the block for many years. And I would say my forte through my career has been in clinical research. In the olden days, which sort of relates to Biomea a little bit, and my talk at Amsterdam is that everything was type 2 diabetes, insulin resistance, insulin secretory defects, hepatic glucose production, and there really wasn't much going on in type 1 diabetes. I was an investigator in the diabetes prevention program, in a few other very large studies, and a bunch of investigator-initiated studies, mostly in type 2 diabetes. And then things sort of shifted, where now we have a lot going on in type 1 diabetes, everything from, anti-CD3 monoclonal antibodies to islet cell replacement, and many other adjunctive therapies. So, the two fields are sort of coming together a little bit. Even the technology is being shared by type 2 now, hybrid closed loop system, CGM. So I would say that is sort of a summary of my background. And being senior, I more overlook some of the younger doctors, younger studies, more recent studies versus being the PI myself. I caught a wicked cold in Amsterdam, so I might have to cough here and there.
Oh, thank you. No, so that's perfect. So I guess, again, from a little more of a broader perspective, what do you think have been the latest trends in diabetes? And as we'll talk about a little later, what are the biggest challenges that clinicians like yourself face when treating type 2 diabetes to start?
Yeah, well, I could summarize the trends in type 1 are hybrid closed loop automatic insulin delivery. That's huge. And at the Amsterdam ATTD meeting, we do have 5 systems available in the United States now, 5. And all the algorithms that drive the automatic insulin delivery, it's all proprietary. They all have their algorithm. But I would say uniformly, it works extremely well, much better than people compared on multiple daily injections doing it themselves, assuming they also have a CGM. Our CGMs are, now we have several CGMs and there's choices. We got the implantable Eversense, and I'm on the board, so I want to make sure I have that disclosure. But then there's, the Dexcom, the Abbott, and probably several more banging at the door of the FDA. So that's the biggest trend in type 1. And of course, we do have teplizumab, that is owned by Sanofi, and they're making a big push for screening for autoantibodies for people in stage 2 T1D. And that's kind of a big deal too, because they can get treated here now and delay the onset of type 1 by, on average, 2 to 3 years, sometimes more, sometimes less. And having type 1 myself, You didn't mention that in my intro, but that's fine. I got type 1 when I was a kid, and I think that's a huge deal because delaying type 1 now in this year by 2 to 3 years, who knows what we're going to develop in the next 2 to 3 years, and to take the stress off young people and older people who get type 1. Type 2, it's, as you know, it's all about GLP-1, GIP, because that class of agents, and I don't want to leave out SGLT2s because they are tremendous in terms of reducing the progression of chronic kidney disease, congestive heart failure. They don't lead to the big weight loss, so they don't get a lot of airtime. But certainly, Ozempic and Mounjaro, the dual agonists, have made a tremendous impact. And look at the indications that they're getting. You just stack them up. Everything from diabetes, obesity, atherosclerotic heart disease, obstructive sleep apnea, pre-diabetes. It just goes on and on. And I do think they have made a big impact, and they will continue to make a big impact. And Novo and Lilly are, they're fighting for the triple agonists and get better products. And so we'll see what happens. But I can tell you, Ozempic and Mounjaro have made a huge impact, and we'll see what future developments bring in that area.
No, that's certainly very helpful, and this would be an entirely separate call or conversation, but see like what the massive impact that potential oral therapeutics in the GLP-1 space could have. But again, that's a separate call. So, as part of my, focus, where we're going to pepper in a lot more of T1D in our discussion, I guess before we look at the recent data at ATDD for Biomea and icovamenib, what do you think is the most misunderstood about the pathophysiology of type 2 diabetes, and what led you personally to focus on type 2 and beta cell biology?
Yeah, well, now that you're asking that question, I didn't really answer your previous question about some of the headwinds about treating type 2, but I could mix this in. Well, as I said, I think the biggest issue with type 2 is it is, and this is something I spoke on, it is a very heterogeneous disease. I mean, we have differences in ethnic differences, genetic defects, familial transmission of defects. So not all type 2 diabetes is the same. And as you know, the folks at Biomea have pointed out that, we have big categories of insulin deficiency and insulin resistance. And that is so true. We have learned in clinic that, certain ethnic groups do not respond the same as others. And I think so people don't realize, I think that's the biggest answer to your question. There's many faces of type 2 diabetes, and that dictates how these patients respond to different pharmaceutical agents. And just a quick note, I'm not sure if you know, but one of my biggest interests through the years has been adherence and persistence with type 2 medications. And I can challenge you on if oral is going to make a huge difference compared to weekly injectable. We can talk about that later. But people with type 2 diabetes have no sense of urgency. I don't want to put them down, obviously. I'm a big patient advocate. But they stop their medications because they see something on television, the copay is too high, and most of the time they do okay. Type 2s can tolerate very high A1Cs, like 13, 14, and not feel too badly. Excuse me, one second.
Yep.
Yeah, and so without a sense of urgency, adherence and persistence is not very good. And when Intarsia came out with their implantable micropump for exenatide, if you remember, that was going to be the big deal because you put the little device under the skin, stays there for 6 months, and adherence and persistence is 100%. But I do feel, and unfortunately, the FDA never gave them the approval, and I thought they really were quite ignorant in that application. But I do think that adherence and persistence has to be one of the biggest problems in type 2 diabetes, by far. You could have the best drugs in the world, but if they don't take them, they don't do any good.
Okay, yeah, that's very helpful. And I guess when you talked about the true heterogeneity of the disease, I guess I would ask first, as you mentioned, Biomea is doing this. I'm assuming the physician community is doing this as well. In focusing on the severe insulin deficient patients, are those patients easily identifiable by you and others? What are the parameters to classify them?
Yeah, that's a great question, Joe. And I think if you look at the guidelines, the ADA guidelines, they don't really steer you one way or the other to help you differentiate at all. You go to the Japanese diabetes guidelines, they clearly say insulin deficient, insulin resistant, and on the insulin deficient side, they recommend insulin and sulfonylureas, but on the insulin resistant side, they don't have that as some of the first options. So, and the Asian cultures, they seem to be more on the insulin deficient side. I always tell the story of I work at the VA hospital for all these years. I always say those vets gave me PTSD working with them, and that's a joke, Joe. There was a lot of Filipinos in the service, recruited from in the U.S. Navy, and they all very similar. They're thinner. They don't have the huge abdominal pannus that the Caucasian type 2s have, and they fail oral agents very quickly. They require insulin much sooner, and they happen to develop complications sooner too. And the severely insulin deficient group typically has a higher A1C, and then higher A1C relates to more complications. So the bottom line is, in practice, Joe, this is an important point. We don't have any guidelines. And it's trial and error. I mean, if you look at the guidelines for type 2, if there is atherosclerotic cardiovascular disease and or chronic kidney disease, you go with the GLP-1, SGLT2. And if the A1C doesn't come down to your goal, then you add the one that you did not add. Then there's categories. If you're worried about weight, then obviously GLP-1s are top of the list and avoiding insulin. So, I mean, it really doesn't differentiate. And how do physicians know? Well, you know what? Experienced physicians, let's say, I would put myself in that category. I may have a hunch if someone's going to respond or not, depending on their age, their A1C, their BMI, their ethnic group, and what have they tried previously. And then, of course, in the Biomea subsets, you can differentiate on add in HOMA B and HOMA R, and there is an online calculator. But to be honest, no one does that in clinic. But maybe in the future, it might help direct physicians to therapies that are going to be more successful earlier on. Then you don't have the trial and fail situation that is not uncommon.
Okay.
Yeah, that was a big mouthful, so sorry.
No, a very helpful mouthful. And as we look to sort of focus now, first we'll start a little bit high level as we talk to, the advancements we're talking about today. What got you interested in menin biology overall? Because it really is a relatively new concept in diabetes. I mean, there have been some correlative studies, and when you talk about the progressive nature of type 2, which you talked about in gauging how and when you give certain therapies, what are you looking for with regard to paradigm shifting therapies such as menin with regard to beta cell preservation and regeneration potentially?
Yeah, well, you're right, Joe. It's a whole new area, and I was attracted to it. I think initially, when I heard that there was data showing that you could preserve lean body mass when you lose weight, and when combined with a GLP-1. And then, of course, the more I learned about it, this could be applicable to not only type 2, but also type 1, a condition near and dear to my heart and my pancreas. And I think the other thing is that's really different is that, it's a covalent binding and you don't need to take this drug forever, at least from the data we know now. You look at the most recent 12 and 24-week data where the A1C continues to drop being off the medication completely. And of course, the newest data at the ATTD was the C-peptide data, which is a clear indicator of pancreatic beta cell production, insulin production. And so it's disease modifying, and it affects both type 1 and type 2. You don't have to take it every single day, which addresses the adherence issue. It's an oral medication. And you know what? The deep down biology of why would our body produce menin? There we don't know everything about this product. I don't. I know that I would say the smart folks at Biomea, Rohit do, but it's a very interesting brand new class with those benefits, and those are huge, I think, and they address several unmet needs in diabetes care.
No, that's fantastic, and we're definitely going to dive into some of the things you teased us nicely with, and I guess the first thing I would ask, even from a more general standpoint, is you mentioned the C-peptide correlations with beta cell function. That's something that's widely known, and ready to be applicable to any data sets that come out.
Well, it is widely known. It's widely accepted, except for the FDA. They do have a stick up their butt. Sorry if I could say this on your call. And the example is with teplizumab, Tzield, the anti-CD3 monoclonal antibody that they have it approved for stage 2 T1D. By the way, they're probably going to change some of the phraseology for type 2 diabetes. Instead of pre-diabetes, they're going to have stages. It sets it up for better research verbiage. But they did I'll make a long story short. They did a study where they gave this monoclonal antibody to people newly diagnosed. That's what we call stage 3. And it clearly showed reductions. It improved the C-peptide. And that's clearly an indication of pancreatic beta cell function. And the FDA has some kind of problem with that. But I do believe they're going to change their mind. They were not accepting of time and range, one of the important CGM metrics. They're lagging behind. But nonetheless, I think any investigator, any person that does clinical research, when you see C-peptide going up when the drug is actually stopped, A1C dropping, that's a pretty unique situation in clinical research.
Okay. Some regulatory progress to be had, it sounds like.
Yeah.
So before we talk about the actual data, I guess my last sort of generalized question is, when you look at the mechanism of action of icovamenib as a menin inhibitor, and right now they're covalent, but some of the others in development are non-covalent. So that's a separate biological debate. I don't know if you wanted to touch on that, but we might or might not. Yeah, exactly. Why do you feel menin inhibition might be more applicable in the severe or insulin deficient or resistant patients we were talking about earlier?
Well, to me, it's common sense. If you're lacking in insulin secretory ability, we'll call that the SID group, severely insulin deficient, and you give something that improves insulin secretion, then I think that's why you saw, and I don't know the data as well as the Biomea scientists, they're excellent. That's probably why you saw the biggest A1C drops, because you're addressing the issue in that group of diabetes. And as you know, the way they classified them was, the age, the weight, the BMI, the HOMA B, HOMA R. So they looked at classic characteristics that you can classify these different groups. And I think they did that trying to figure out where's the best place this medication fits. A1C, I think I missed that. A1C, weight, age, and HOMA R and HOMA B.
No, for sure. So I guess now is when we're going to talk about about the actual data, and you definitely alluded to some of this earlier. So they showed in these insulin deficient patients achievement of a 1.47% reduction in HbA1c at week 26. And this was 14 weeks off of therapy. How surprising is that to you for a diabetes therapy? How good is this reduction, the magnitude here in this kind of patients? And I keep repeating it, the focus that,
Yeah.
these data were achieved while off therapy for quite some time.
Well, gosh, you're feeding me a good question. It's well, put it this way, it's unheard of. When in diabetes, you stop the medication, as I said earlier, talking about adherence and persistence. When you stop the medication, oh my God, it, the A1C starts going up, it stops working. And that's why adherence is such a big deal. So to have continued improvement while off therapy is, I'd say, very unique. I've never seen it before. I would challenge anybody else that said they did. And so that is very, very, very unique. And then the 1.4, of course, that's significant. Gosh, anything over zero point 0.5, but 1.4 is, very significant. And, I think they definitely need to do more patients. It was only phase 2, but it's a pretty darn good start and very exciting to think that this might be a real thing when you give it to a larger group of people.
Okay. And I guess, these are relatively early data, even though they're really compiling, a decent amount of patients. I guess I would ask a little bit of from the field type of comment or question, when you were at ATD, how some of your colleagues, reacted to the data. I can't ask you to talk about the investment reactions because I think a lot of investors are still wrapping their heads around, the mechanism of action here because they're so used to seeing what you just described. Here are the current therapies, you stop, you go back up. So, what kind of reactions were you seeing?
Yeah. Well, I don't really follow investors' reactions to be honest.
Exactly. More from the field of the physicians.
Guys,
That's
you
all.
guys are all crazy. You overreact
Yeah.
and you panic. But I will acknowledge that you guys are very smart and a lot about diabetes. Well, I think the reaction I don't want to sound too simple, but it's the same as mine. Is that how could you get a reduction in A1C when you're off therapy for that long? That is very unique. Then at the same time, you have increase in C-peptide, which validates the mechanism of action. And it's also a disease modifying condition. And I would say, a lot of the people in the audience might be, when we look through the categories that the Biomea folks put together, primarily to help understand the data, the SID, the MARD, we want a little dance, the 4 different groups. It makes perfect sense. And in fact, we've been classifying insulin resistance and insulin deficiency for a long time. We just never had any formal acronyms for it. So the reaction is the same. They've never seen a medication where you stop it and it continues to work. And then I think the wheels were turning that, wow, what a great way to address the poor adherence and persistence we see in the type 2 population. And that problem is huge. You look at the A1C, the NHANES database over the past decade, and even add 15 years, despite the advancements in over 55 type 2 meds, not all different classes, but repeat ones. I remember the DPP4s were big, and then the TZDs, the SGLT2s, the GLP1s. And why is the A1C straight across? And I know that I published on that, and that ended up in the Biomea, one of their slide decks. And it's one of my passions which is why I run this not-for-profit, Taking Control of Your Diabetes, because try to motivate people to understand the importance of taking their medication, getting involved in their care, getting a home glucose monitor, and hopefully a continuous glucose monitor. And the type 2 population is a tough group. Once again, they don't have a sense of urgency. Us type 1s, we do have that sense of urgency. So it's an important area, and I think their reactions were the same as mine, because we don't have in the States ways to categorize which way you start or stop medications based on insulin deficiency or insulin resistance. We do know how the drugs work, for sure. So, we eventually get there. And I would say has to be trial and error sometimes.
Okay, okay. And I guess with regard to, I guess the disease modifying properties and why it's different, I guess the first question I would ask regarding this is what's also completely different than the current treatments is that it appears that icovamenib would not be chronically dosed, which is, totally apart from what you would see today with other therapies, and that would potentially increase patient adherence. I guess it's somewhat of a rhetorical question, but maybe get some of your reaction to that.
Yeah, no, it's it it it's hard to wrap your head around because we've never had anything like that. And you talk to anybody that sees patients in clinic or talks to physicians in clinic, the type 2 top population is tough. And there's no medication that I know of that once you give it, it works. And if you stop it, these folks continue to do well. Because even with GLP-1s, as you know, you stop Ozempic, you stop Mounjaro, people gain the weight back, even though they try super hard to keep it off. And I've had a couple patients that said, I can do it. And sure enough, I had someone yesterday come crawling back asking for a refill when they gained their weight back. But it's not just the weight. Those drugs, have protective effects on the heart, the kidney, et cetera. And you lose those protective effects as well. And of course, as I mentioned earlier, the other thing that I think should not be forgotten, I mentioned it once, is that when these folks yo-yo on these GLP-1 type drugs, they lose weight, they lose fat and muscle, but when they gain weight back, it's all fat. So you're putting them in a worse situation. And the data with icovamenib is pretty interesting on preserving muscle mass. I'm not an expert in the details of that, but I think that data is extremely interesting and really sets them up to be a nice partner with people on GLP-1s.
Okay.
/GIP.
Sure. No, that's very helpful and good perspective, obviously. I guess it might be early, but it's, still provide some thoughts here. I mean, things could change tomorrow for all we know. We talked about C-peptide correlations. Your views right now, albeit potentially early, that if we're impacting beta cell biology, restoration, what have you, what are your views of the potential, I'm using all my temperate words carefully, the potential lasting effects of this therapy if it's not chronically dosed?
Well, time will tell. And I think it's very. I'm hopeful that because of the way this drug works and the covalent binding and what I've seen so far, it looks good. But I think we have to wait.
Absolutely. Completely fair answer. And, from what I've seen, and of course you've seen more here, any comments with regard to safety and tolerability of the drug?
You know what? I reviewed that data last night and it looks really good. Nothing really sticks out. So I even think the FDA hold on the type 1 study was premature, but they're here to protect our research subjects, and as you know, that hold is released. So it looks pretty safe. There's no red flags at this point, as far as I can see in the data that I looked at last night on the clinical trials.
Right. And so that's all extremely helpful. And I guess this would also be a topic for another call, but do you have any initial commentary with regard to combinations going forward if you were to, say, combine icovamenib with the GLP-1s, for example?
Yeah, no, I think that's awesome. And in fact, I think it could even make the combination more potent than either one alone, which is sometimes you don't see that when you add things together. So you have a GLP-1/GIP that works on, glucose-dependent insulin secretion, glucose-dependent glucagon suppression. And I think that if you have a drug that allows the beta cells to be more responsive, I could see that the combination being very effective. And because there's autoregulation going on with the GLP-1s and when I say glucose-dependent insulin secretion and glucagon suppression, I don't expect to see more hypo at all, just because they work together. So you know what? I think it's it's very interesting that the way icovamenib works, and then it might be just a really physiologic combination for those two to be put together.
Okay, yeah, time will tell because Biomea is doing some early, studies on
Yeah.
that. So we'll see how they play out. Right now, at least the preclinical data. I don't know if you wanted to comment on that, but the preclinical data definitely look promising.
Yeah, no, nothing more to
Yeah.
add. It does look
No,
promising.
absolutely. So I guess, I said you didn't want to just sort of like pepper it in here because I think it's important as the drug's profile expands. Did you want to discuss its potential in type 1 diabetes at all from a high level perspective?
Well, I'd say from a very high level, if icovamenib could help improve insulin secretion, remember that when you get type 1, your beta cells are destroyed by the autoantibodies. But with better assays, we have discovered that even when someone goes to the older assays to measure C-peptide or insulin, previously when it was undetectable, with the newer assays, you actually can detect C-peptide years after the diagnosis of type 1. But I would just saying that the earlier, if it did show beta cell function improving, then I think the earlier you intervene, the better, and it could be used as stage 2 or even stage 3. So I think it has potential for sure to be an adjunctive therapy, probably not replace insulin, but adjunctive therapy is where we're at in type 1, and that could be a very good pharmaceutical agent, especially, you give it oral once a day with your insulin. And if you can preserve the remaining undestroyed, beta cells, you might have a longer honeymoon period. And anytime you're in the honeymoon period where the beta cells can secrete insulin, someone's control is much better. They have less variability, more time in range, less time below range. So anything we can do to improve that period, anything to improve our honeymoon period, not with our three ex-wives, but
Yeah.
a joke. No, I think that it has potential. But I think once again, the potential to have preserved beta cell increased C-peptide in early type 1 has a lot of potential.
Okay. Yeah. Again, looking forward to playing out there. And I guess if we just go backwards a little bit here, with regard to type 2, for example, and the combinations with GLP-1s, I was remiss not to bring this up, but you mentioned one of the issues. If you get off a Mounjaro, for example, and you bounce back, it's all fat. You've lost the muscle. Some of the early human islet cell data and animal data in combination with GLP-1s with icovamenib have shown preservation of muscle mass. So I don't know if you could talk to those data and also even anything out there in the field that are looking to help protect muscle mass with the GLP-1s.
Yeah, well, you know what? Once again, you outlined it quite nicely. The animal data looks very good, and I think I can't predict the future, what's going to happen when you get it in humans. But I think the way the drug works, and that it makes sense that the lean muscle mass is preserved. And all I can say is it just needs to be studied. I'm sorry, I can't add too much more than that. But I think it's a very unique property, and that's what I answered earlier on. That what got me interested a year ago at the ADA in Orlando when I saw some of the data on lean body mass preservation, and that's huge. That's the big downside of GLP-1s. As you know, adherence is not very good for a lot of reasons. People can't pay for it. It wasn't available. They see something on television once again, they cycle up and down. That's just not good. That's like someone being, on a many different diets and yo-yoing their weight. So, there is such thing as physiologic lean body mass. When you're 300 pounds, let's say you lose 100 pounds on Mounjaro, you don't need as much muscle mass to walk around. So there is some natural loss of muscle mass when you lose weight, but nothing than what we've seen in people who cycle.
Okay. Okay. And I guess sort of coming back to a little broader question, if icovamenib or, just the menin concept, as, more companies look to menin inhibition here for diabetes, how what is the current status right now? I mean, you're a key opinion leader in this area. Other centers of excellence in diabetes might be more amenable to the impact of menin on beta cell positive impacts, in pathophysiology of beta cells and restoration or what have you. For the broader diabetes physician community, what do you think the learning curve would look like if it progresses to more advanced stage studies and even approval?
So you're asking me, physicians out in the clinic seeing patients?
Yeah.
Well, I think it would be taken on very receptive, mainly for the reasons we both mentioned already, that a finite duration of therapy, continued success. I mean, that's like, especially nowadays where access to clinic is so poor. I don't know what happened after COVID, but to get an appointment every 3 months is basically impossible. Every 6 months is somewhat doable, and that's all over the country. So if you can give someone a medication that is going to continue working after, 12, 14 weeks, that is such a great clinical advantage. And I think, that, when you have data combined with GLP-1, because more and more physicians, even primary care doctors, are using these GLP-1 drugs, mainly driven by the patients who see all these movie stars losing weight. But it's such a big win to put those two together. So I think it would be very well accepted. There's really no negative downside that I could even think of. The side effect profile is good. It's oral medication once a day. So I do think that, you have a finite duration of therapy. It's actually something that they've never seen before, but I think it'll be easy to educate these doctors, to be honest. I think this is the kind of therapeutic advance that's going to get their attention. No question about it.
Okay, fingers crossed on that.
Yeah.
And you know what? I think my last question will come back to one of the problems you described earlier, and that's on the regulatory front. You mentioned a huge dichotomy with regard to Asia and the US FDA in defining patient populations.
Yeah.
Is there any ongoing progress right now at the FDA to help with these patient definitions? Are they working on any guidelines or guidance for the industry? Where does it stand, or is it still sort of a black box?
Well, the ADA guidelines, I think, have improved quite a bit compared to the years in the past. Because in the past, they just said, start with metformin first, and then they had, 4 different options, and then you pick one, and then the third level is pick something that you haven't used already. So we are more disease oriented, atherosclerotic heart disease, kidney disease, obesity, and fear of hypoglycemia. And there's one other category, I believe, but I think so. They have advanced. And I think as drugs like icovamenib gets out there, and I'm sure the ADA will be looking at where would they put it on their treatment algorithm. I think it has to get a little closer, phase 3 clinical trials. But I do see the guidelines being flexible. And that's one thing that has improved over the past couple of years. And that they adjust depending on what therapies are out there. So it's possible that they will be able to put in a category and maybe even suggestions on how to determine if a patient is more insulin deficient, like the Japanese Diabetes Association. So in that the different ways to calculate the SID and the insulin resistant group and the 2 other groups, there's a publication on Lancet, and there's an online calculator. So it is quite easy. We use online calculators all the time to, BMI on a very simple level, but FIB4 to pick up fatty liver disease is used all the time now. Sometimes it's built into the hospital laboratory data. Like, you order a chem panel and platelets, and then it knows the patient's age and duration of diabetes, and comes out with a calculation. So I think I think it's really a good thing. I think the icovamenib advancements is going to help us look at the different subgroups of diabetes, type 2 diabetes that we treat. And I think we can be a lot more successful. So it's kind of an icebreaker in that regard, Joe.
Okay, no, that's definitely good to hear, despite your tones about the FDA and jokes, because
Well,
you know,
you know what? They're good. They protect us. Sometimes
totally.
they're really quite slow.
Yeah, I know. What
Okay.
can we do? But at least we're seeing progress along all different avenues. But look, Dr. Edelman, this was extremely helpful. A lot of great information here, but not only about the space, but hopefully the promise of icovamenib moving forward. And also, I was also neglectful to state also from a full disclosure standpoint, I do cover Biomea with a buy rating, so that's been disclosed now as well. Thank you very much for your commentary, and I look forward to the progress of the drug and also to see how your participation increases with that as well. So thank you.
Joe, thank you very much. I've enjoyed speaking to you and whoever's listening, and hopefully we'll meet again.
Absolutely.
All right. Bye.